talk now. I've just got off a rather long video call with Dr. David Speaker, the renowned Canadian virologist and analytical scientist. And I'm going to run through the main findings of what we talked about, and then you can watch the video in detail if you want all the nuance. So just a quick sketch really through what is we talked about on the video. Now, Dr. Speaker's analysed 32 vials of RNA vaccine so far, and has found DNA contamination in all of those vials that is analysed from different countries. And in the Pfizer vaccine, he's found something called SV, simian virus 40 enhancer and promoter. More on what that does in a minute, but basically it means that the DNA from the contamination can get into the nucleus of the cell and into the DNA of the host cell. The DNA is found is residual plasmid DNA from post-process impurity. So this DNA from E. coli bacteria, the sort of bacteria you've got in your colon, this impurity should all be taken out, but it's not. The manufacturing process has left it in Pfizer and in even larger amounts in the Moderna vaccines from the E. coli. Because viruses, of course, can't grow on their own. They can't make their own energy, so they've got to be cultivated in some sort of cell culture. And the cell culture used in the manufacturing processes is E. coli, that they're the sorts of cells, the bacterial cells, that the virus is brewed up in. There's free broken up DNA. So the DNA is partly broken up. So it's the E. coli are lies. They're broken up and the DNA and the RNA comes out. The RNA is what the manufacturers want, of course. But there's also DNA comes out as well. DNA fragments come out. But the thing is, this DNA is packaged into the lipid nanoparticles. So the DNA that's not supposed to be there is packaged into the lipid nanoparticles with the RNA, and that means it's systemically distributed around the body because these lipid nanoparticles go everywhere. That means the DNA from the post-processing impurity goes to the cells potentially all around the body. And that basically, the fact that it goes into the lipid nanoparticles guarantees that the DNA fragments get into cells. Now, the limits of DNA contamination that are allowed in vaccines were really formulated before the lipid nanoparticle technique was developed. So small amounts of DNA going into your arm won't do too much harm, but when that DNA is in lipid nanoparticles and it goes everywhere, it's a carrier mechanism to take it around the body. There's the systemic biodistribution. And also that means that the lipid nanoparticle ensures the entry of the DNA contaminant into the body cells. So it really is quite a problem with guaranteed systemic distribution. Now, work from 1999 has shown that 3 to 10 copies of these DNA fragments, this SV fragment particularly, 3 to 10 copies getting into a cell can facilitate transport of that DNA contamination into the cell nucleus, as long as the cell is not dividing. If the cell's in what we call the interface, when there's no active cell division going on. So only 3 to 10 copies of DNA contamination can cause that contamination to go from the blood, from the lipid nanoparticles where the blood takes it to the cell, into the cytosol, the cytoplasm of the cell. And 3 to 10 copies means it can go into the nucleus of the cell. And once it's in the nucleus of the cell, what have we got in the nucleus of the cell? Well, we've got our own DNA, our own deoxyribonucleic acid, and it can be incorporated into that. When the foreign DNA gets into our own DNA, that is a change in our DNA. And when you have a change in the DNA or a change in the genetic material, that is defined as a mutation. And mutations can lead to malignant change. So integration would mean mutation. We know that that can happen. Now, cells in culture exposed to the lipid nanoparticles contaminated with the DNA. So human cells have been cultured, exposed to this contaminated vaccine, the RNA vaccine with the lipid nanoparticles. And when the host DNA of those cells has been analysed, it's been found that the DNA from the DNA that's contaminating the vaccine does get into the chromosomes of the human cells. Now, this has been demonstrated in cell culture. It's not been demonstrated in whole people, but there again, governments aren't looking for that. So let me just explain that a bit again. So they've made cultures of human cells. So you have a culture of human cells growing in a dish or something. You then add the vaccine, the mRNA vaccine, which contains the DNA contamination. You then let that brew for a period of time. You let it incubate for a period of time. Then you take the DNA from the host cells, human cells in culture. You take that DNA, and the DNA that contaminated the RNA, the DNA, the contaminant that was in the lipid nanoparticles, does get back into the DNA of the host cells. This has been demonstrated. So we know this happens in cultures, in tissue cultures. Now, what we need to do, of course, is take people, for example, who have long vaccine injury, take their cells, do a complete genomic analysis, and it will be a relatively simple matter to demonstrate whether this foreign DNA was in their cells or not, but that work is not being done. If you want to know why that work is not being done, I don't know, ask a politician. They might know why it's not being done. So, and the chromosomes that have been contaminated, for want of a better word, the chromosomes are made of the DNA and some additional proteins called histone proteins. And chromosomes 9 and 12 have been demonstrated to have been contaminated with the DNA from the contamination from the lipid nanoparticles in the so-called RNA vaccines, which I guess we must now call RNA/DNA vaccines. And I've got a note here, cancer tumours a year post-vaccine. So basically there can be a delay before cancers form, I think is what I mean from that note. So the mutation's been proved in an oncogene. Now an oncogene is a gene which can stimulate mitosis. And of course cancer is an out of control mitosis. So we know that at least one oncogene, one potential cancer causing gene in chromosome 9 or 12 can have been altered by this DNA. So the theoretical risk is clear. And to me, that theoretical risk is enough to stop these DNA contaminated vaccines until we know one heck of a lot more about it. But we know enough to stop it now, unequivocally, in my view. Health Canada has said there's no increased risk of cancers. And they also said at first that the Pfizer vaccine does not contain SV40, but they were wrong, it does. They later said that although the SV40 is present, so a bit of backpedaling there from Health Canada, that the SV40, the SV40 is the contamination sequence, one of the contamination sequences of DNA that can get back into the nuclear material of the cell and cause mutation. Health Canada says that's got no functional role. So first of all, they said it's not there. Then they said, oh, you know what, it is there. Then they said, but don't worry about it, it doesn't do anything. But then Health Canada asked Pfizer about the residual fragment. So having Health Canada declared that it does nothing, they then wrote to Pfizer. So Health Canada know that this contaminating SV40 potential cancer-causing sequence is there. And then Health Canada was asked, like a Freedom of Information request, Health Canada was asked for this information from Pfizer, but it came back redacted. Why would they want to hide the scientific information, Health Canada? Why would they want to do that? Now, Dr. Speaker uses a technique called fluorometry. Now, this basically attaches a fluorescent molecule to the DNA. And then that means when the DNA is present, you can see it with your microscope. It fluoresces and you can actually see it. It's giving off light. Now, this is a good technique. This fluorometry is a good technique to give quantitative analysis of all of the DNA. So it analyzes all of the DNA that's present. It's a better technique than quantitative PCR because it's analysing all of the DNA and labelling with this fluorescent marker all of the DNA that's present. And it's found that in some DNA, in some RNA vaccines, there is 10 trillion copies of this DNA sequence per dose. Now, I was taken aback by this. Dr. Speaker has found that the Moderna one particularly, can contain up to 10 million copies of DNA fragment per dose. And we've just said that 3 to 10 copies of SV40 DNA fragment is enough to facilitate transport into the person's own DNA, into the cellular nuclear DNA. So 3 to 10 copies per cell and up to 10 trillion copies. So that means that potentially 1 trillion, that's 1,000 billion body cells, could be transfected with foreign DNA. And that just, I was taken aback by that. So 10 trillion copies of DNA contamination potentially present per dose, 3 to 10 copies enough to cause incorporation of SV40 DNA contamination into the nuclear genome of the cell, thereby causing a mutation, which has been identified in cell cultures in chromosomes 9 and 12, including an oncogene that can potentially cause cancer. 10 trillion copies per dose, or up to that.